Hi, everybody. I'm Jane Wilkinson, and I'm the president of CANCollaborate. And we're a new nonprofit that is focused on rare cancer collaboration. And just like our friends here at Travera, we also grew out of an idea at MIT. So there's a real kind of, synergy and a connection here. So at CANCollaborate, we're really focused on creating a new platform and a new way to think about rare cancers. And initially, our focal point is going to be with appendiceal cancers. Now, appendiceal cancers are extraordinarily rare. About 10 in a million people will experience an appendiceal cancer diagnosis. And what we hope to do here at CANCollaborate is to develop and optimize a truly collaborative model that brings together leading researchers, oncologists, surgeons, and, with our friends here at Travera, real innovators and real technologists. So our model is designed to consolidate the expertise, facilitate data sharing and model sharing. And also, when we talk about model show, this is about talking about sharing samples as well. We really want to create a more comprehensive and more collaborative approach to appendiceal cancer research. So within CANCollaborate, we have created a network of appendiceal cancer CANCollaborators. They are oncologists, they are surgeons, they are researchers, they're advocates, and importantly, they also are patients too. And through this collaboration that we have with Travera, they are all going to have access to the Travera technologies alongside the Travera community of partnerships, which include many patient advocacy and many research groups. So with that, I'm going to pass it over to my colleague at Travera, Rob, and he can tell you more about how this technology works.
Thanks, Jane. My name is Rob Kimmerling. I'm the CTO, one of the co-founders of Travera, and I'm going to give a quick overview of our cell mass measurement platform and how we're monitoring drug response in live tissue specimens that are sent to Travera. So in terms of the background for the type of measurement we're collecting, it's a very similar basis to functionalized precision medicine generally, where the idea is to test drugs directly on a tissue specimen from a patient. And the reason for this is currently the main approach for functionalized or precision medicine is genomics-based, molecular readouts from tissue specimens or patient samples to match drugs with those patients to see what might be most effective. The only issue is that this relies really on very well-established drug mechanisms. There's only a few contexts in which this has been very successful. And there's a fair number of patients that unfortunately don't benefit from this type of genomically informed precision oncology. And so functional medicine is directly measuring tissue against drugs and trying to fill this gap, trying to find drugs that will work for a larger fraction of patients that are going through treatment. There's a couple of key advantages to this type of approach. One is broad applicability. So in contrast with genomics-based precision oncology, it's agnostic to drug mechanism. This works across a broad types of therapies. Another benefit is that we can directly test a patient's own live tumor cells. So this, instead of population-based precision oncology, this is a truly functional, personalized approach for this type of readout. And then finally, it's applicable to any stage of disease. So in contrast with genomics, again, rather than only measuring a specimen upfront early in therapy, we're able to test this type of approach several times throughout a patient's course of treatment, meaning for late-stage patients that have relapsed on prior treatments, we're still able to test their tissue against a panel of drugs to see which might be working well. So Travera takes a pretty unique approach to functional precision medicine. We are actually relying on cell mass measurements to see which drugs are having the greatest effect on a patient's tissue specimen. This is built around a platform called the Suspended Microchannel Resonator that was developed in Scott Manalsa's group at MIT. And what this is a platform that allows us to take very highly precise measurements of single cell mass. So the way this works is this is a microfluidic device with a vibrating cantilever that's built into the platform. And we are able to pass cells in suspension across this sensor. And as these cells pass across the sensor, we see a change in the resonant frequency of that cantilever, how fast it's vibrating. And that change in vibration is directly correlated with the mass of that individual cell. And so by measuring that mass readout and linking it with a downstream image, what we're able to do is characterize the morphology and also the mass of each particle that's passing through the sensor. This is important because for a primary specimen that's a very heterogeneous population of cells, we're able to get a nice clean distribution of cell mass measurements. And it turns out that cell mass is actually a very highly functional proxy for the state of a cell, the phenotype. And it's very integrated for the metabolism and growth of each of these cells, and specifically how it's responding to a different therapy. And so what we can do is we can treat cells ex vivo with these different drugs, and monitor their mass response with these different distributions, comparing both the absence of drug and the presence of drug, and then look directly at how each one of these drugs is affecting the distribution of cell masses. What that lets us do is actually get a statistical measurement of how strongly a population of single cells is responding to a therapy. And this mass measurement is really what we're using as the functional outcome of the platform. And because this is a single measurement of mass response and really just a proxy for how the tissue is responding, it's, as I mentioned, pretty agnostic to the drug mechanisms that we're looking at. As long as the drug has a cell intrinsic mechanism, we're able to back out whether or not that drug is having an effect on the sample. So we've now been able to validate this approach across a wide panel of drugs, or over 100 FDA-approved therapies that we validated this test for. And what we found is that mass has the ability to resolve response in all of these drugs and do so more quickly than standard measurements of something like viability that you might grab from cell titer glow or a flow cytometry readout. The reason that's important is because it allows us to characterize the response of these tissues in a very short time frame. What this means is that when cells are taken ex vivo, they begin to die just naturally by virtue of being outside of their natural environment. But we're able to characterize mass response or drug response with these tissues very quickly and actually get a sense of whether or not they're responding prior to the cells dying naturally. And with this in mind, we've now validated this large panel of drugs that lets us test primary specimens with this same readout. So here we're just showing an example of what this type of mass response readout looks for a large panel of drugs. This is 20 drugs that we're measuring from a pleural effusion specimen from a breast cancer patient. And what you can see is we're looking at a wide array of both cytotoxic and targeted inhibitors in this case. And this is the main output that we would see from our assay that we're running at Travera, which is looking at a mass response score on the y-axis here. We're essentially giving a score between 0 and 100 as to whether or not a population of cells is responding to drug. So if that score is above 50, we say that is a statistically significant response to that therapy. And if it's below, we're seeing that there's not really an effect that the drug is having on that population of single cells. So we've validated this approach for a wide panel of primary specimen formats at this point. We've done this for solid tissue specimens, including core needle biopsies, fine needle aspirates, resections. We've also done this with hematologic malignancies, with blood and bone marrow specimens. And in general, the assay itself is fairly agnostic to what the tissue format is. Once we get down to a pure population of single cells, which is part of our CLIA assay, we then run that same mass response measurement regardless of the specimen source. So today, we now have this full workflow where we're looking at drug response measurements, and we're able to link these responses to patient outcome for that same population. We have this for about 60 patients now. So for these patients, these are drugs that we tested on primary tissue taken ex vivo. And then that patient received this therapy, and we're able to track how they responded to that treatment clinically. And what we found is for both positive and negative predictions, we're sitting right above 80% in terms of our ability to predict how a patient will respond to therapy. We've done this across a range of solid tumors and leukemia, multiple myeloma. And one of the benefits of this data set is actually including both positive and negative predictions. So this is mostly an observational work that we've done so far, but that does give us a chance to test this negative predictive capacity as well. Another key detail of this data set is it's in a very heavily pretreated population of patients. So these are patients that have seen 3 or 4 lines of therapy previously, and we're still able to conduct this measurement and have this pretty high predictive accuracy, which gives us confidence that our test is actually being used to determine the outcome for the cells that we're measuring. This is really the bulk of what Travera is focusing on now, is really building out this clinical evidence. And the main instrument we have to do this is our CLIA workflow. So with the platform that we've developed from MIT, we've sort of built this CLIA workflow around it. And this is the heart of the company at this point, where this main workflow involves collection and shipment of a tissue sample. These are in specific kits, depending on the tissue format, whether it's a solid tissue or a malignant fluid. After overnight shipping to Travera, we purify single tumor cells from those specimens and then plate them in various conditions that we're going to measure downstream. We then use our panel of instruments in the lab. This is about 16 that we have running right now. And we collect these linked measurements of both imaging and single cell mass to construct the mass response readouts that we're having for each one of these conditions. After we compare all the mass distributions for each of these conditions, we're able to then put together this test report, which actually describes which drugs induced a significant effect on that particular patient's tumor cells. One of the key benefits of this workflow is that the turnaround time is just 2 days. So because we're only dosing these cells for 1 night, it's really 1 night for the shipment to Travera and 1 other day for us to actually run the assay with return of results that evening. So this is the one thing we're quite proud of, is this ability to turn results around very quickly and sort of stay in line with the necessity of guiding therapy if this test is going to be used for that. Another benefit is that we can actually conduct this assay with a small number of cells. It's only about 5,000 cells per condition that we need to make a call as to whether or not a drug is working. And so this allows us to work with low input specimen formats, such as fine needle aspirates, where we may only end up with 100,000 cells or so, but we're still able to test a panel of drugs and return them in the report. So this workflow is really our source of collaboration at this point. We are trying to get specimens into this workflow and generate as much data to just have clinical evidence showing accuracy and predictive accuracy moving forward. And this is really a place for partnership that we're most excited about. So with that, in terms of the details of collaborating and how we work with Travera, I'll turn it over to our VP of Business Development, Dennis Watson, who can explain more about that process.
Thank you very much, Rob. And as Rob mentioned, we're excited about our early data and technical applications, but we are looking to expand upon them. And we want key collaborators and researchers to contact us with that thought in mind. We're already doing some early work in HIPAC with a number of institutions, and we're looking at other ways to expand upon that, as well as many other applications that make sense for a therapy guidance technology like ours. One of the nice things about being a CLIA approved lab is that while those research collaborations and partnerships are being established, we have a unique ability to allow you to run the test in clinical scenarios to gain some firsthand experience, to identify the key process and flow and patient type that's going to work in your institution and in your specific study environment. And we can do that without the effort of having to set up an MTA or formal research agreement. So the way that you would accomplish this is simply by contacting me. My contact information is on the screen now. Once you reach out, I like to have a few days notice to be able to get a test kit to you ahead of any planned procedure. I can work with you and your staff through completing the order form that CLIA requires, walking through the selection and priority of the drug list that we'll analyze for your patient, and then just collecting and sending the sample. Once we receive the sample, we'll process it and return results to you in about 2 days, all through this CLIA approved pipeline. Again, this gives a great opportunity to get some firsthand experience with the test while more formal partnerships and agreements are being established. We're doing this at no cost. We're not going to invoice the hospital or you or the patient in any way. We're not going to bill this to insurance or Medicare. We're running these cases at no cost because the value of that experience, understanding that process and flow, is just as important to us as it is to you. And so we're excited to be able to work through this with you, and I hope to hear from many of you that are potentially viewing this presentation. I want to thank Jane and the team at CANCollaborate for the opportunity to be here today and to expand on the exciting partnerships that they've already helped us to establish. So I want to just toss this back to Jane, and thank you all very much for your time.
Rob and Dennis, thank you so much for that overview of how the technology works and how you're able to work with our CAN collaborators, with our partners, with our oncologists, with our surgeons, and importantly, also with our patients. I think this is a great opportunity to learn a lot more about the disease and to really have an impact in patient care. So thank you both, and thank you for working with us here at CANCollaborate.